So before I talked about the cell cycle and the processes, the actual activities of the cell cycle, and I talked briefly about the checkpoints. But I think I didn't talk as deeply enough and as in detail as I wanted to. So in this video, I'll thoroughly explain the regulation of the cell cycle. So let me move and let me zoom out a little. Let's see this. So there could be regulation by external factors such as a growth factor, a stimulus, a hormone, a protein, etc. And all of them could make positive activation. So they could, let me show this. So let's say we had a cell in the G1 phase, and then we had this was, let's say, a protein or some hormone or something that positively affects it. Just do a little text stream there. That, and protein, and the protein, let me get a little bit less. Protein. And with that protein, it could proceed into S phase. This is positive regulation. And it also could be that it could be negative. This is positive. Now, so now, if we have the same situation, the cell wants to move from the G1 phase, this is the G1 phase, to the S phase again, but maybe something stops it from moving into the S phase. Let's say it's a CDK. A totally possible scenario, a CDK could inhibit, and this is called inhibit. So this inhibits S phase. This, from the G1 to the S, it inhibits the S phase. Or there could be regulation by internal checkpoints. So let me zoom out a little so that you can see it. The whole diagram. We have an interphase. We have this big sliver. That, until G1. And at G1, there's something. So, and then this is S. This is G2, and this tiny sliver, like 10%, or taking like about 1.5 hours. That is the mitotic phase. That is the actual mitosis. Let me scroll through. We have 3 checkpoints. We have a G1 and a G2 and an M checkpoint. Now let me go back on this diagram to draw where they are. So about at the end of the G1 phase, I'm putting this line over here. And then it could proceed, or it could go into a non-dividing state. It could go into a non-dividing state. And that non-dividing state is called G0. Sometimes the G0 state could be, sometimes permanent. Sometimes permanent. So sometimes permanent,
or sometimes so that I'll have to trash the stick box. Sometimes temporary.
And you might be thinking, an example, please? Yeah, you might be thinking for an example. So, sometimes permanent, that could be nerve cells. So that nerve cells. So some nerve cells, actually some, not all nerve cells, some nerve cells. Let me scroll through. Some nerve cells. And sometimes temporary. That could be liver cells. So when did they recover out of G1? They recover out of G1 when they have injury. Oops, I did some things wrong. Injury. Whoa, did something wrong there. Injury. Okay, so now let me scroll back to this point. Boo, that isn't right either. The G1 checkpoint. Whoa. G1 checkpoint. It checks if the cell is large enough to divide. And this is really, really important. So important that I'm going to circle it. Cell is large enough to divide. This is really, really important. External positive stimuli. So you could have some stimuli over here, here, and here. So I'm going to label positive was a plus. And they check for sufficient resources. So it fails, it tries to fix, but usually it goes to G0. This is what we talked about. If it succeeds, it goes into S phase, which is synthesis phase. So we also have, let me clear out some of my dusty things over here. Now, let me begin by underlining the G2 checkpoint. It checks if DNA has properly replicated during S phase and if cell is large enough to divide. Oops, I circled too much there. Cell is large enough to divide. If it fails, then it tries to fix the problem in quite a few ways, actually. Completing DNA replication is one way. Repairing damaged DNA. Oops, damaged DNA. Let me change that. Damaged is a D. Damaged DNA. Restarting the cell cycle is another way. And if problem is unfixable, triggers apoptosis. And apoptosis is programmed cell death. And it is made by cell communications. And there's a very important molecule that participates there, which is called P53. That is really, really important. Actually, it is so important that Science Magazine made a Molecule of the Month in 1996. And P53 is more important than even chocolate. Imagine. Yeah, so Khan Academy said that, imagine, like, Science Magazine made this Molecule of the Month in 1996. And the theobromine, which is the main chocolate molecule, has not earned this award before. So, whoops, something's going wrong. No worries. So if it succeeds, it proceeds to mitosis, and this is important. That sounds new. Look at the cell cycle video, but that's really, really important. This is division of the nucleus and not the
cytoplasm. It is not the division of the cytoplasm. No, I don't want to put this in this text field. I want to put a different text field. So let me start up a new text field. This nucleus. Nope. No, I want to put it somewhere else. No, I want to put it somewhere else. Okay, nucleus, or I could put cytoplasm, and I'm going to cross out cytoplasm. Cytoplasm. Crossed out. No cytoplasm. So let me, if it succeeds, it proceeds to mitosis, which is the division of the nucleus. Let me clear all this thing. Let me clear all that. And let me eliminate the word cytoplasm here, because it actually is not right. I've just labeled it to cross it out. Whoa. Seems like I'm having a little trouble with the text thing. Cytoplasm. So we have the important M checkpoint. And also, like, kinetic nodes? What? Like, did the computer autocorrect some things? Kinetocores. I didn't even notice. This is what really happens in mitosis. I don't know why. But the computer autocorrected this, but the right word is kinetocores. Really, I'll underline this. Are attached to spindle fibers in metaphase. If it fails, it tries to fix the problem, and it usually doesn't restart the cell cycle. And if it succeeds, it continues into anaphase. And once it goes into anaphase, I'll label this like this. It cannot be reversed back into metaphase. So this is a one way. This is one way, not the other. Okay, so let me begin by clearing out all the clutter here. Oops. So we have this image here. So this is a much better diagram than probably what I drew. Whoa. And first of all, let me also begin by clearing away everything I drew before. Be it a stem cell. No. Mitotic phase. And here we have this G1 checkpoint. G1 checkpoint is cell growth. So this is cell growth. And there's this D1 checkpoint, which is restriction. And after this continues, this is a very important detail. That big part continues. It is irreversible. So it is completely irreversible, so it is not reversible at all. And let me begin by erasing that. So once it continues, it is irreversible. So sometimes there are textbooks that mark there is an S checkpoint. I saw this in Biointeractive. So sometimes you need to think sometimes there is an S checkpoint. So they sometimes label here. So S checkpoint. I haven't talked about it before, but HHMI Biointeractive, I've seen that it also labels S checkpoint. And let me take this away, and then we have a G2 checkpoint. And in the mitotic phase, we have an M checkpoint, which is actually a metaphase checkpoint. It should be better called metaphase checkpoint. And formation of two daughter cells.
So now I actually have another image. The second image. OpenStax. Cyclic expression cycles. So cyclic expression cycle. So you have a lot of cyclins. One of them is called cyclin D, and you can already see cyclin D. Cyclin D. I'm highlighting the path. This is cyclin D. And cyclin D is most active at around S phase. That is probably around the S checkpoint. So that is probably, let me abbreviate this thing by putting just an S there. And cyclin D is pretty constant, but it's most active during S phase. Now cyclin E, cyclin E, let me highlight this part, is cyclin E. It is active at around G1 phase. So cyclin E is active during the G1 checkpoint, G1. Cyclin A, let's look at cyclin A. Let me highlight cyclin A. I also begin by highlighting cyclin E in a different color by not confusing you with things. It's not as confusing. Cyclin E there. Now I have cyclin A. That cyclin A goes around what? Goes around G2. That is ground 2. By the way, it's called ground 2. And let me highlight cyclin B. This green line, this green curvy line that I've drawn here is about the same. There. And that is around M, or metaphase. So now, that is the cyclic expression cycle. That is really, really important. That is really, really important. So we have, and let me go back to this because let me also begin by drawing the cyclins in there. So here at around this G1 checkpoint, we looked at this back again. Let me draw an arrow around G1, around S, around G2 at this G2 checkpoint, and around metaphase at M, mitosis. So we looked at that cyclin E is most active during G1 phase. This is cyclin E. During S phase, the most active cyclin, is cyclin D. Cyclin D is also pretty constant compared to all the other ones, which abruptly drop down. This is cyclin D. There's cyclin D. And we also have cyclin A, which is G2 phase. Cyclin A. And then during mitosis, we have cyclin B. Cyclin B. And then we have formation of the two daughter cells. And the G1 checkpoint, and let me put in parentheses, irreversible. And it's not really there, so let me just put a little text field there. A little text field around the cyclin E. Can I just draw a little better? Let me just put this on this screen. Put it around here. Irreversible parentheses. And that means that this action of cell division during the G1 checkpoint cannot be reversed, and that's why you need to be really, really careful after beginning to divide. So let me go back to the next image. By the way, I have a lot of images from OpenStax. And the next one is about RB regulation. And we have this unphosphorylated RB which doesn't have P's attached to it. So let me label
that no P's are attached to it. P, I label 2 of those, PP, and then let me do a crossed out P, crossed out P, crossed out P. And crossed out P's just mean no phosphorylation is going on. And it binds transcription factor E2F, and E2F is actually labeled there. E2F, it cannot bind the DNA. Cannot bind the DNA. Because what happens is that the cell is not growing. So the cell is not growing. Let me show that. And then cell growth means the cell becomes bigger. This cell becomes this cell. And that is what triggers the P's to come back and to become active. Let me also put that in the right place. I don't think I put it in the right place. Bigger cell, a little bigger. And this is what makes this ATP to ADP. So these cells have a lot of ATP. We want ATP. And phosphorylated RB, it releases E2F. So releases E2F, which binds the DNA and turns on gene expression. Makes a transcript and turns on the gene expression. Then it comes into S phase, or it could turn on M phase. It could turn on two things. So now let me go to this image. Let me get a little less to make this all fit. So now let me go to the second part here. Dun-dun-dun. I have to zoom out on there too. Zoom out there. When the G2 checkpoint fails in a normal cell, well, first there is going to be a fix of the damage. You should try to fix the damage. One fix of the damage. And if this is not possible, no fix occurs. And then it continues DNA replication. Now I put a 2 there. Third, it could try a cell cycle restart. Or if the damage is unfixable, it triggers apoptosis. So this S, big mutated S, turns into normal S. If this is not possible, then it triggers apoptosis. Okay, so now we have this important molecule and an important image, by the way, about P53. Remember I told that P53 was the molecule of the month in 1996. And this is really, really important. So let me zoom into that image. And we have a normal P53. It could have DNA damage, cell cycle abnormalities, or hypoxia. And P53 is active, so P53 is not broken. And it could cell cycle arrest, and or it could make apoptosis, or it could start DNA repair and restart the cell cycle. So until the damage is repaired, so until we have this mutated S becomes normal S, so until this happens, the cell cycle is arrested. So the G1 phase cannot continue into the S phase. So until this, it's not going to happen, right? But with mutated P53, P53 is broken or malfunctions, or maybe there is no P53, no P53, crossed out P53. The cell cycle
continues and the cells can become cancerous. So this happens. So the G1 phase could continue into the S phase or whatever phase, goes to whatever phase it needs to. That is the progression of the cell cycle. And the old S, the mutated S, does not become the fixed S. And it could become cancerous. Cells could have cancer. And cyclin-dependent kinases. Let me go down here. Let's say there is a lot of this CDK. So an excess of CDK here and there. This shows that there are 4 pluses instead of 1 plus. That is an excess of CDK. So excess. And there is an excess of cyclin and an excess of CDK. And there is a complete excess of kinase as well, a lot of kinase. And the cyclin CDK complex is phosphorylated, and there is a ton of ATP, a big mass of ATP, CDK, and cyclin. There's an excess of ATP, an excess of cyclin, and an excess of this CDK. And this target protein, there's an excess of protein as well. There's a ton of excess. So excess ATP, excess CDK, excess cyclin, and even excess protein. And because there is an excess, what could become is that there could be a ton of cell division. So there's a lot of cell division. Chun, da, da, da. And all these arrows are going to lead to excess cell division. So let me draw that. Let me scroll through. Done, 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 done. All these get together. Done, 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 done. And they all become lots of cell division. And lots of cell division, and it could become cancer. Lots of cell division. Division. So lots of cell division. So let me actually draw division by showing that. That becomes 2 of these. Lots of this. And sometimes it is unnecessary. A lot of this unnecessary cell division. Let me zoom out there. Lots of cell division that is unnecessary, and it actually could lead, this scenario could lead to cancer. Isn't good at all. Lots of cell division that is necessary, and let me just do this. So now we're going back to this image, this RB regulation of the cell, and this unphosphorylated RB, it binds the transcription factor E2F. But let's say we have mutated RB. So mutated RB, and let's say it is hyperactive RB. So maybe what could happen is maybe you could have, let's say, hyperactive E2F. Let me show hyperactive by this So with no RB,
the E2F could bind to the DNA and it could start transcript. So it could start the transcript already without this happening. So with no cell growth could continue hyperactive. And this is actually how important cell regulation is. Cell regulation is extremely important in that case. Like if you have hyperactive RBs, let me just zoom out to show all the scenarios we've been talking about. Non-functioning or mutated P53, excess ATP, CDK, cyclin proteins, or hyperactive RB. Each of these are bad scenarios for the cell. Each of them are actually bad scenarios. And these all demonstrate, all of these are actually mutations. Let me draw this big arrow. These are all important mutations. These are really all mutations, by the way. So these are all mutations. Mutations. Let me draw mutations. That's how important these all are. Because non-functioning, these are all mutations. And since you have all these mutations, then what is going to happen is really bad, because all these mutations affect a part of the cell cycle. So if there is excess protein or excess cyclin, even that scenario is not really a mutation, but it could lead in excess cell division, and it could become cancer. But P53, this molecule of the month award, is so important. It shows how important P53 is. Because otherwise, the DNA gets damaged, the cell cycle isn't normal, and hypoxia, and the cell cycle continues without repair, which means that they could become lumps or even benign tumors, and at some point they could become malignant tumors, and then at some point they could even become these, they could even metastasize, and they could get out. So they could get out of their part of the body and infect other parts of the body. So, like cancer, that's why cancer is so difficult to cure. And that's why all these issues are really important to know about, and they're really important. Because like even hyperactive RB could lead to excess cell division and cancer. And if there is a mutation actually, in this cyclin pathway, then it might be even worse. And because you have all these mutations that are accumulating, it may break the DNA replication scheme. They may even break DNA replication and make mutations more frequent and make cancer happen more easily. So this is how important having P53 and all the things you need is so important. It's so important to have P53 and everything in your body properly working. Thank you. Bye.